This is the introductory lecture for a class on antibiotic resistance given in the winter session in alternate years at the Harvard Chan School of Public Health. In this introduction, I'll cover the dimensions of antibiotic resistance, a bit of the history of antibiotic resistance, some aspects of the biology of antibiotic resistance, some social and behavioral views of resistance, and some guiding questions and concepts. I'll mention at the beginning that I'll use the term antibiotic and antimicrobial interchangeably. Technically, an antimicrobial agent is any chemical that selectively kills bacteria, while an antibiotic is an antimicrobial agent that is produced by a living system. Many of the early antimicrobial agents were antibiotics produced by species of bacteria or fungi to kill other microbes. We will use both terms to mean antimicrobial agents, whether natural or not, throughout this video. When we think about antibiotic resistance, we can identify several dimensions. The first is which kinds of bacteria are being considered. The second is whether we are considering bacteria that are mainly transmitted in the hospital or in the community. Next, we can think about the antibiotic class that is involved. A class of antibiotics is a set of chemically related drugs that work by a similar mechanism to kill or inhibit bacteria. For example, beta-lactams like penicillin and amoxicillin, or macrolides like erythromycin and azithromycin. We can think about the genetic mechanism of resistance. That is, how does the bug acquire the genes that make it resistant to the antibiotics? We can also think about the biochemical mechanism, that is, what is the bug doing to become resistant to the antibiotic? When we think about how antibiotics select for resistance in bacteria, we can think about the different mechanisms by which they do so, first in individual patients who are treated, and also in populations of hosts. In the full version of this class, we spend a lot of time thinking about study designs to assess these mechanisms. Pathogenic bacteria, that is, bacteria that cause disease, can be divided into two broad biological categories, gram-negatives and gram-positives. As you'll see in a minute, these two groups of bacteria are very distantly related to each other, so it is often the case that a particular antibiotic works only in one of the two groups, or works much better in one of them than the other. Also, there are some broad generalizations about how these different types of bacteria tend to transmit, so discussions of the problem of antibiotic resistance often focus on gram-negatives or gram-positives, especially in recent years on gram-negatives. The terms derive from the different reaction of these two different types of bacteria to a simple staining procedure that can be carried out in almost any laboratory. Gram staining involves use of a pink stain followed by a purple counter stain, and the gram-negative bacteria are those which do not emit the counter stain, while the gram-positives do. As a result, under the light microscope, gram-negatives are pink, while gram-positives are purple. Escherichia coli, often just called E. coli, is perhaps the prototype of gram-negative bacteria, and certainly the best studied. Gram-positives include the anthrax bacterium and Staphylococcus aureus, among many others. As you can see on this tree of life, with eukaryotes in red, archaea in green, and bacteria in blue, the bacteria are by far the most diverse organisms, and the gram-positives are quite distant from the gram-negatives, exemplified by the proteobacteria at the bottom. Mycobacteria, while technically gram-positive, are evolutionarily quite distinct from other gram-positive pathogens, and are often considered as a third separate group. The most important mycobacterium from a medical perspective is Mycobacterium tuberculosis. Now, a bit of the history of antimicrobial resistance. Concern about antibiotic resistance arose even in the earliest days of antimicrobial chemotherapy, as in this article by Paul Ehrlich in The Lancet in 1913. In it, he described two principles which are still today considered to be important principles for prescribing antibiotics. Because he was a German writing in an English medical journal, he naturally used Latin and French to make himself understood, so I'll translate. He said, "The great sterilizing therapy consists in this, that by means of one or at most two injections, the body is freed from the parasites. Therefore, the old therapeutic remedy is applicable: hit hard and hit early. He goes on to say, a further advantage of combined therapy is that under the influence of two different medicines, the danger of rendering the parasites immune to arsenic, which naturally would be a very great obstacle in connection with further treatment, is apparently greatly minimized. Thus, in this early article, he describes the idea that we should treat as early and with as high a dose as possible, and one should treat with multiple drugs to prevent the emergence of what we would now call resistance mutations. These two ideas are central to modern strategies of treatment, though the hit hard and early idea has been recently subject to some criticism. This slide, created by Tom O'Brien from Brigham and Women's Hospital, shows a timeline of our continuing struggle to find antibiotic drugs that effectively treat those gram-positive and gram-negative pathogens. The blue arrows show the introduction of different classes of chemical compounds, such as sulfa and penicillin. The hatched lines in pink show periods in which there were certain gram-negative strains of bacteria that were resistant to all drugs currently available. Similarly, the purple lines show the same thing for gram-positives. As time progresses, new classes of drugs are introduced, and new resistance mechanisms, shown as the boxes on the arrows in purple and pink, pop up and become obstacles to effective treatment. Alexander Fleming, who discovered penicillin, noted very early on that resistance was inevitable and emerged very rapidly within the laboratory when he tested penicillin against certain bacteria. Writing about a synthetic class of antimicrobials, the sulfonamides, which had been invented before penicillin, he noted, soon after the sulfonamides came into practice, it was discovered that some strains of what were generally sensitive microbes were resistant to their action. This could be due to one of two things. The sensitive organisms might have been eliminated by treatment with the drug, while the insensitive ones persisted and were passed on from one individual to another. Or that by insufficient treatment with the drug, sensitive microbes might have acquired resistance or fastness to the drug. There is probably no chemotherapeutic drug to which in suitable circumstances the bacteria cannot react by acquiring fastness. For very many of the antibiotics and species of bacteria, it only took a few years between the introduction of antibiotics and the appearance of the first resistant strains. This table shows the timing of discovery, clinical use, and first resistance, and as you can see, it takes as little as less than a year, or more typically a few years, before resistant strains start to appear. Of course, the fact that there is some resistant strain of some species around does not mean that the drug becomes useless, only that testing becomes necessary before one can be assured that this drug is useful. Also, there is one very important exception to the rule that resistance follows quickly from antibiotic use. Penicillin resistance has never been seen in group A Streptococcus, the cause of strep throat and some more severe diseases such as necrotizing fasciitis. This remains a mystery, and several different hypotheses for why it might be were reviewed in a paper now almost 20 years ago. To my knowledge, we still have many hypotheses and no certainty about why this has happened. Once resistant strains emerge, they do not become the majority overnight. It takes some time for resistant strains to increase in frequency in the population under the selective pressure of antibiotic treatment. Typically, that timescale is several decades or more, as shown in these graphs, of three types of resistance in some gram-negative, hospital-acquired pathogens. For some other infections, like the gram-positive hospital-acquired infections shown here, spread can be quite gradual. Another puzzling observation is that the prevalence of resistance of different antibiotics often seems to level off over time, reaching a plateau in a given population well below 100%, though often high enough to preclude the use of the particular antibiotic for that infection unless susceptibility has been demonstrated by laboratory tests. I say this is puzzling because when we have made mathematical models of transmission of resistant and sensitive strains, it is very easy to make a model which sends resistance over time up to 100% or keeps it at zero, but it's in fact quite difficult to make a model where the equilibrium frequency of resistance is intermediate. Another clue to the underlying dynamics is that if you plot the annual usage of antibiotics, in this case penicillins, in different European countries on the x-axis, and the prevalence of resistance in an organism like Streptococcus pneumoniae on the y-axis, you see a positive correlation. As interesting as the correlation itself, you might note the variation between countries in the amount of antibiotics they use. France, on the right, uses about 4 times as much antibiotics per capita, focused on penicillin, as the Netherlands, its next-door neighbor, shown here at the left. Although recently, France has embarked on a relatively successful campaign to reduce overuse of antibiotics. Similar correlations are seen in the United States, with the Southeast and Mid-Atlantic using the most antibiotics and having the highest prevalence of resistance. So that's a little bit about how resistance goes up in populations, but what about the decline of resistance? Once resistance is common in the population, can we go back again? There are some situations where the answer is probably yes. This is an outbreak of gentamicin-resistant, methicillin-resistant Staph aureus in a neonatal intensive care unit in one hospital. Around a quarter of all infants in the unit were infected with this multi-resistant strain, and following the implementation of a bundle of control measures, including antibiotic control, cohorting, and isolating those with the infection from those who didn't, the prevalence went down to 0 in a few months. Here's the epidemic curve of another outbreak, which distinguishes between imported cases in gray and transmitted ones within the unit in black. You can see a spike in transmitted cases following several imported cases around the middle of the graph. And in response, there was a strengthening of infection control, shown by the purple. Although introductions of this resistant bacterium continued, transmission seems to have been halted, possibly by the enhanced infection control measures. While there is some success stories like this in individual hospital units, with community-acquired infections it has been much more difficult to control the level of resistance. This slide shows the massive change in the use of sulfonamides, a class of drug that is often used to treat urinary tract infections in the United Kingdom between the late 1980s and the 1990s. The major use of sulfonamides was in the combination treatment trimethoprim sulfamethoxazole, also called cotrimoxazole. As shown in the open circles, you can see it was reduced nearly to 0 over that period. However, the prevalence of resistance to sulfonamides remained nearly stable, going from 39% in 1991 to 45% in 1999. This slide shows data from an intervention study in Alaska, where there was an effort to reduce antibiotic use by educating people about the fact that antibiotics are not always required for certain conditions. On the left-hand side is the situation in 2 regions, an education region and a control region that did not receive education. Bars show the prevalence of intermediately and fully resistant strains of bacteria. Again, this is Streptococcus pneumoniae resistant to penicillin. Results seemed very promising in the first year, as resistance declined in the education region but stayed unchanged in the control regions. However, in the second year of the study, the prevalence of resistance remained stable again in the control regions, despite the fact that they now received their first year of education. And it went back up to baseline in the region that was receiving its second year of education. This was not a very encouraging study, as you can imagine. In spite of these depressing examples, it is clear that resistance in some community-acquired pathogens can go down when antibiotic use declines. A way we can see that is from data on the seasonal variation in antibiotic use and the concomitant variation in resistance. On the left side here is a graph of prescriptions each month of various kinds of antibiotics in Israel, and you can see a pronounced seasonal variation on the log scale. There is about a 2 to 3-fold decline in prescribing during the summer, and an increase back up by the same amount every winter, with no dramatic trend in overall prescribing across the years. On the right side is the prevalence of antibiotic resistance in isolates of strep pneumoniae from the middle ear of children, also by month, but now combining the different years. Whether we look at penicillin resistance, erythromycin resistance, or multi-drug resistance, there is evidence of a seasonal cycle with peaks around December or January and low points around August or September. Take a minute to think about how we could reconcile these two kinds of observations. On the one hand, major interventions that seem to have little effect, and on the other, repeated seasonal changes in the prevalence of resistance every year as antibiotic use changes. The next major topic I would like to discuss is the biology of antibiotic resistance. First, how do antibiotics work and how do we measure resistance? When thinking about how antibiotics kill, it is important to note that in fact, some antibiotics don't kill at all, but only prevent cell division. These are called bacteriostatic drugs, and they often work by preventing protein synthesis, for example. Others kill bacteria directly and are called bactericidal. Bactericidal drugs often kill only dividing bacteria because they inhibit processes that occur only in dividing bacteria. This makes treatment of latent infections, or of bacteria that are dividing slowly or not at all, sometimes very difficult. Tuberculosis is a famous example. A few years ago, it was shown that bactericidal drugs have a common mode of action, regardless of which process they directly target in the cell. The ultimate death of the cell results from a common end process involving reactive oxygen species. Treatment of a patient with an antibiotic often requires help from the immune system of that patient. This is especially true with bacteriostatic drugs, which can't kill the bacteria but only prevent them from dividing. In this case, something is required to clear those bacteria so that they don't begin dividing again once the antibiotic pressure ends. There is also a phenomenon known as persistence, or phenotypic resistance, that occurs in many bacteria. This graph shows on a log scale the death of bacteria under antibiotic treatment with ampicillin in vitro. You can see that the number of bacteria goes down rapidly because ampicillin is bactericidal, but plateaus with 100 or 1,000 bacteria per mil left even after 6 hours of treatment. Here again, it is believed that the immune system is required to mop up what the antibiotics can't kill. This may help to explain why it is often found that resistant strains of bacteria emerge more often in immunocompromised patients than in immunocompetent ones, because even if there are only a few resistant bacteria, they are able to grow in the absence of an effective immune response. How do we quantify resistance in bacteria? The level of resistance is defined as the minimal inhibitory concentration, or MIC, of the antibiotic for the bacterial strain. A strain with a low MIC is called highly susceptible, while a more resistant strain has a higher MIC. A very common way to measure this is by broth microdilution, where around 100,000 bacteria are placed in each of several wells containing different levels of antibiotics and incubated for a period of time. At the end of incubation, the wells are scored as growth or no growth, and the lowest concentration of antibiotics that prevents growth is defined as the MIC. This can also be measured by diffusion of the antibiotic from a strip on an agar plate, where the area in which growth of individual colonies is inhibited can be read as a way to quantify resistance. Often, clinicians do not want to know the exact MIC of a strain, but only whether it is sensitive or resistant to clinically achievable concentrations of an antibiotic. Therefore, committees of microbiologists establish breakpoints separating susceptible from resistant, sometimes including also intermediate resistance. Sometimes there is a smooth variation from susceptible to intermediate to resistant, as in the case here of penicillin in Streptococcus pneumoniae in Japan. In other cases, there is a strong separation between susceptible strains, most of which have an MIC less than or equal to 0.06, and resistant strains, which are 50 to 100 times more resistant as shown by this middle peak, or even as much as 2,000 or more times as resistant as shown by those that are over 128. We should note that measuring the MIC itself is a simplification, because bacterial growth typically is slowed and then becomes negative as the concentration of antibiotic increases. This graph shows that the decline in growth rates can follow any of several different shapes, and the MIC is really a one-parameter summary of the whole curve. Therefore, in understanding resistance, it's important to realize the MIC has some limitations as a measure of resistance. First, it ignores bacterial subpopulations within a population of bacteria that may be variable in their susceptibility to antibiotics. In effect, if you measure the ability of the population to grow on a certain amount of antibiotic, you are measuring the MIC of the most resistant strain within that population. Also, the MIC depends on the conditions under which the bacteria are growing. These include pH and availability of nutrients. So the MIC in a petri dish or flask may be different from that in human tissue. Finally, as mentioned before, there are different inhibition curves shown at right that all have the same MIC. Considering that concentrations of antibiotics often fluctuate during treatment of the patients, those different curves may have different implications for the effectiveness of treatment, even though they all show the same MIC. Next, let's discuss mechanisms of resistance. When we talk about mechanisms, it can mean several different things. We could be talking about the genetics of resistance, how it's encoded. As we'll see, certain types of resistance can be transmitted from one bacterium or another, depending on the genetic element that carries them. So by genetic mechanism, we mean what is the medium on which the instructions are written to become resistant. Second, we can ask about the biochemical mechanism, which is what exactly is the bacterium doing to become resistant. Finally, we could talk about the mechanisms by which resistant is selected and spreads within populations. How do our actions increase or affect the frequency of resistant bugs? First, genetic mechanisms. One genetic mechanism is intrinsic resistance. Sometimes every member of a species is resistant to an antibiotic and always has been. For example, tuberculosis is resistant to penicillin because it naturally encodes a beta-lactamase, an enzyme that breaks down penicillin. Vancomycin is an antibiotic that is produced by another bacterial species. The species, of course, has to be resistant to the action of vancomycin, and in fact, the genes that confer intrinsic resistance on that species may turn out to be the progenitors of the genes that confer resistance on some of the pathogens we treat with it. Another form of intrinsic resistance is failure to activate a prodrug. A prodrug is a chemical substance that has to be modified before it becomes active as an antibiotic. Two of the major antibiotics used to treat mycobacteria, isoniazid and pyrazinamide, require alterations by enzymes in mycobacteria before they become active as antibiotics. Other bacteria do not have such enzymes and thereby are not sensitive to these drugs. This slide shows some of the other ways, besides intrinsic resistance, by which bacteria can become resistant to antibiotics. The first is by mutation, where some part of the chromosome of the bacterium, shown here as a circle, changes, perhaps by a single nucleotide change, to encode resistance. The second mechanism is by plasmid transfer. A plasmid, shown as a smaller circle inside of a bacteria, is an accessory element to the main genome of the bacteria, which carries genes that may not be essential in all circumstances, such as antibiotic resistance genes. Not all strains of bacteria, even within a particular species, carry the plasmid, and the plasmid may be transferred from one individual bacterium to another, either another member of the same species, or even, in some cases, members of a different but related species. A third mechanism is transformation, in which DNA released, for example, from another dead bacterium is taken up actively and integrated into the chromosome of the living bacterium. Again, this may occur between or within species. These different mechanisms have important consequences for the behavior of resistance in populations. Mutational resistance implies that it is very easy to get a resistant strain in almost any patient infected with some organisms, because mutation frequencies are around 1 in 10 million to 1 in 10 billion individual bacteria or viruses. In a large population, they will therefore often be present. An exception is if multiple mutations are required to confer resistance, as then the frequency of the multiple mutant will be lower. Examples of pathogens where all known resistance comes by mutation are tuberculosis and HIV. Other mechanisms may be less likely to emerge in a single patient because they require DNA from outside the organism. However, these mechanisms allow the evolution of very complicated ways of resisting antibiotics because they can move multiple genes as a block from one bug to the next. Sometimes this is even across species. A rather amazing phenomenon is the existence of so-called epidemic plasmids. These plasmids can spread from one bacterium to another, crossing into multiple different species, causing an epidemic of drug resistance within the bacterial population. On the left are epidemic curves of drug-resistant bacteria from multiple species of gram-negative bacteria such as Klebsiella, Serratia, E. coli, and Enterobacter. On the right is the restriction digest or molecular barcode of the plasmid extracted from each of these patients. Although the host bacterium was quite diverse from one patient to the next, the plasmid was identical or nearly so across multiple patients, as you can see from the similar restriction patterns. There are quite a number of other mechanisms by which resistant bacteria can share genes encoding drug resistance. Just to mention one more, an integron is a modular system by which multiple genes coding resistance to one or more antibiotics can be snapped into a DNA cassette, which is itself often carried on a plasmid, and packages these genes together in such a way that they confer multiple resistance. There's a lot more to say about the genetics of resistance, but let's move on to discuss briefly the biochemistry of resistance. If you think of a bacterial cell as trying to resist the onslaught of a chemical that is going to interfere with some target inside it, bacteria have evolved almost every possible strategy you can imagine for how to avoid that. They can reduce the permeability of the membrane, thereby keeping out the antibiotic. They can express efflux pumps that pump molecules of the antibiotic out. They can degrade the antibiotic by breaking it down within the cell or in the periplasm around the cell. They can detoxify it by adding a chemical group to it, such that it no longer acts on its target. They can express an enzyme that alters the target itself that the antibiotic is coming in to reach, for example, the ribosome or the machinery of cell wall construction. They can alter that target directly by a mutation in the gene encoding it. They can make more of the target to swamp out the action of the antibiotic. Or they can inactivate the enzyme that activates the prodrug so the antibiotic can enter but does not become toxic to the cell. Each of these strategies has been observed in nature. Most of these mechanisms are quite specific to one drug or class of chemically related drugs. For example, Enzymes that alter either the drug or its target are specific to the class of drugs that have a given structure or attack a given target. Similarly, changing or amplifying a target molecule only helps against those drugs that share the target. There are a few mechanisms that confer resistance to more than one class of drug, perhaps the most important being an efflux pump, which is sometimes quite broad in the substrates it can pump out of the cell. These are sometimes known as multi-drug resistance transporters. Depending on the mechanism, a bacterium may be rendered highly resistant to essentially any concentration of the antibiotic that could be clinically achieved, or it might just become partially resistant with a small increase in MIC. Now let's talk about mechanisms by which selection for resistance happens. A key concept for understanding how antibiotics select is to understand that we have a normal flora of bacteria that colonize multiple sites in and on the body, like those shown here. With the possible exception of the lungs, each of these sites contains some bacteria, even in completely healthy people. While some of these bacteria are completely harmless under all circumstances, many of the most important pathogens that can cause severe infections are often common colonizers of the skin, respiratory tract, or gut, and when they remain there, they cause us little or no trouble. I have listed some of the major ones here with dark blue for gram-positives and red for gram-negatives. When we take an oral antibiotic, nearly all of these populations will be exposed to it and experience selection, even if they are not responsible for the infection we are trying to treat. This creates the phenomenon of bystander selection, whereby the treatment of one infection can cause selection for resistance in other populations of bacteria at different sites and of different species. This helps to explain why there is so much concern about avoiding unnecessary use of antibiotics. If there was no infection there, the antibiotic can't do any good, but it can still do harm by selecting resistant strains of bacteria in the normal flora, which may then cause disease in the same individual and be transmitted to others. Indeed, in many situations, much of the harm done by use of a drug may be to species other than the target of treatment, such that the optimal dosing to cure the patient may not be optimal to prevent resistance. Looked at from the perspective of the bystander, most exposure of a given species to a given drug may be due to treatment of other infections. I think it's useful to describe three mechanisms by which antimicrobial use increases antimicrobial resistance. The first is what's known as acquired resistance, selection of resistant strains within a host. In this mechanism, a patient is initially infected with a susceptible organism, but upon treatment, a resistant variant is selected and outgrows to become the majority population. A second mechanism, which is probably at least as important, but is a bit more subtle, is primary resistance, or selection within the host population. Here, primary resistance means that a patient is infected with the resistant organism in the first place. But the way that antibiotic treatment promotes this is that it reduces the transmission of susceptible infections by clearing the susceptible bacteria. Thus, a patient who is exposed to potential infection from many other patients is more likely to get infected by resistant bacteria that have not been cleared by the treatment of these other patients. Finally, treatment can increase the susceptibility to colonization of a resistant strain that you didn't have before. By clearing out some of the normal flora, treatment may open an ecologic niche so that a person can become colonized by bacteria through transmission, some of which will be resistant. As with genetics and biochemistry, it's important to think about which selective mechanisms are active in a particular case. Acquired resistance during treatment is crucial for many viral infections, for tuberculosis, and for Pseudomonas, another bacterial infection, as well as for a few cases especially related to the class of drugs called fluoroquinolones. This mechanism of resistance creates a negative relation between treatment success and selection, because successful treatment prevents the emergence of resistance within the host. A major way to ensure successful treatment is to use adequate doses and combinations of antibiotics to prevent the emergence of resistant strains. For primary resistance, on the other hand, which probably applies to almost all bacterial infections, the success of treatment at eliminating the susceptible strain from one host is the very thing that creates selection for resistance in other hosts by making transmission of a resistant strain more likely, as susceptible strains are less prevalent. And for changes to susceptibility to colonization, which probably is important for many of our normal and commensal organisms, such as Streptococcus pneumoniae, and for many nosocomial or hospital-acquired pathogens, we really don't understand the relation between selection and treatment success, because treatment is directed at one population and selection is occurring in another. Another important phenomenon is that of co-selection. The idea here is that if we exert selection in favor of a particular strain of bacteria by whatever means, all the properties of that bacterium will increase in frequency, as they are part of the same genome. A particularly memorable experiment was performed by Ann Summers a few decades ago to explore the hypothesis that dental work promoted mercury resistance genes, and that these were linked to antibiotic resistance genes. Summers hypothesized that installing and removing dental fillings could promote antibiotic resistance. Indeed, when she did these experiments on monkeys, she found that mercury resistance increased over time following the procedures, and that it brought with it resistance to tetracycline and ampicillin. That is because the same genetic element in the same bacteria carried resistance to both. While that's a particularly striking example that I like to describe, perhaps more relevant in the clinical context is that bacteria resistant to one antibiotic tend to be resistant to many antibiotics. That means that treatment with any one of the antibiotics to which a strain is resistant will tend to promote resistance to the other antibiotics within the population. This is a table of correlations between resistance to different antibiotics within this bacterial species Staph aureus. It's not important to look at every entry, but the stars denote statistical significance, and you can see that some of the correlations are quite strong, in the neighborhood of 50 to 70% to levofloxacin with several other drugs, for example. This slide shows data from the early 2000s on resistant strains of gonorrhea in men who have sex with men in the United States. Resistance to ciprofloxacin, a fluoroquinolone, increased over that time period, mostly on the background of resistance to penicillin and tetracycline, shown as the black triply resistant line. Those two drugs had previously been used to treat gonorrhea. That meant that the use of ciprofloxacin tended to increase the prevalence of resistance to those two other drugs too, so that you could not easily use those older drugs to treat strains that were resistant to ciprofloxacin. In a recent review, Xiaohan Chang from our group laid out several hypotheses for why multiple drug resistance seems to cluster in the same strains. Lastly, let's spend a few minutes thinking about antibiotic resistance from a social and behavioral perspective. Public health agencies like the CDC often promote the notion that if you use more antibiotics than you need, it will be bad for your health. There are some cases where this is probably true. For example, if it makes you more likely to get infected by your own normal flora, which are now likely to be resistant following treatment. Or there are studies suggesting that perhaps antibiotic treatment contributes to childhood obesity by altering the microbiome, although that's been challenged recently. When thinking about resistance and its solutions, it is helpful to think whether the individual patient may be harmed by their own antibiotic use. It's at least as important to think about the externalities of antibiotic use, that is, the effects on others in the population when a particular individual uses antibiotics. We can think of susceptibility of bacteria to drugs as a finite resource that we use up to some extent when we take antibiotics. Or we can think in terms of a related notion that antibiotic use is like pollution that may provide some benefit to the polluter but at cost to society. Such notions fall under the broad umbrella of the tragedy of the commons, described in a classic article you should read if you haven't, by Garrett Hardin in Science in 1968. He describes a commons like the Boston Common on which people graze their cows. He notes that it's in everybody's interest to use more and more of the commons by grazing more cows. But if everyone follows that self-interest and takes as much as he or she can, soon overgrazing will occur, and there will be no resources left for anyone. Perhaps a more intuitive image is the one here with pigs eating from a trough. Each one wants as much as it can get and benefits from using the resources, but deprives the others thereby. This graph, which we saw earlier, makes the point in the context of antibiotics. People in France may gain something from using lots of antibiotics, perhaps more rapid relief from mild infections like ear infections. But they do so at the cost of making antibiotics less effective for themselves and their fellow countrymen. I should mention once again that France has recently embarked on a reasonably successful effort to reduce overuse of antibiotics. Much of the problem of antibiotic resistance on this view is driven by selfish behavior of drug companies wishing to sell more product, prescribers wishing to complete encounters with their patient more rapidly by giving the patients a prescription, farmers who use antibiotics for growth promotion and to counteract the effects of intensive farming on animal health, and patients who often demand antibiotics when they don't really need them. From the evolutionary perspective, this creates an arms race, where bacteria become resistant to the latest antibiotics, and we keep having to improve the antibiotics or discover new ones in order to keep up with resistance. As we have seen, that is not always easy to do, and the supply of truly novel antibiotics seems to be slowing, leading to significant attention to the problem in 2016 on a scale greater than I have ever seen before. A different approach is to see the problem as one of social norms and behavior change. Here the idea is that antibiotics are overused, and this is really to no one's benefit. So the goal is not to induce people to be less selfish, but to change the social norms of what is beneficial. Also, measures such as infection control, wider use of vaccines, and other interventions may help to change the balance by reducing the amount of antibiotic use and thereby decreasing resistance. However, many behavioral interventions on antibiotic use have failed, and it's not clear even when they succeed that resistance is substantially reduced by lower use. These different perspectives suggest different kinds of solutions, although many people would argue that all types of solutions are needed as the problem is multifaceted. We could try to change user behavior by education or by modifying norms, or we could regulate our use of antibiotics, making it harder to get them, for example, in the hospital by requiring consultation with an infectious disease physician before prescribing is permitted. Some have even suggested taxation, though health insurance makes that a difficult solution to implement. But the notion of mutual coercion mutually agreed upon is the solution that Hardin proposes to tragedies of the commons. There's been a lot of work recently trying to stimulate innovation in antibiotics by changing the economic incentives to make new ones. And of course, part of the solution is to prevent infection by hygiene, vaccines, and other precautions. I'll close with what I consider to be some of the outstanding concepts and questions in the study of antibiotic resistance. First, it is clear that the phenomenon is an interesting one both scientifically and from a public health perspective. But how bad is it, and how bad will it get if we don't change course? Recent estimates place the dollar value of $100 trillion on the cost of antibiotic resistance over the next few decades. Some might think that is a rather speculative estimate, but how should we measure the costs, and what should we compare them to. What can we do for an individual patient to prevent resistant infections? This is a fundamental question of medical care, hospital administration and systems, and clinical epidemiology. Why have some types of drug resistance increased quickly and others slowly or not at all? And why have drug-susceptible strains persisted in almost all species despite high levels of antibiotic use? What can we do in the population as a whole to reduce the risk of resistant infections? Here the tools might include education, vaccination, hygiene in the community, or other interventions. Finally, what is the relationship between doing the best we can for the patient and their treatment, and the effect of the burden of resistance in the community? Does the best treatment for the patient increase the burden of resistance in the community, reduce it, or perhaps both to different extents over different timescales? That concludes this brief introduction to the biology and epidemiology of antibiotic resistance.